Hello everyone. Now we are going to discuss about the cellular and molecular hallmarks of cancer. Cancer cell show some special properties or acquire some special properties. They help them to grow in a big mass by evading various immune mechanism and process of cell death, apoptosis. They grow in a big mass and they develop certain kind of altered metabolic adaptation, so they become a big mass and invade the surrounding tissue and result into the metastasis. So what are these properties? First is self-sufficiency in growth signals. They do not need additional growth signals. They basically will result due to the mutation in proto-oncogenes. They overexpress and result into the uncontrolled or excessive cell proliferation. Second is insensitivity to growth inhibitory signals. Cancer cell render insensitive to growth inhibitory signals and further result into the more and more cell proliferation. Third property of cancer cell is evasion of apoptosis. As we know, the cell with damaged DNA or some other irreparable defects will result into the death of cell by apoptosis. But cancer cell acquire some special feature to evade the process of apoptosis. Fourth property is altered cellular metabolism, also known as Barber effect. As the developing cancer cell need numerous intermediates or macromolecule for the increased requirement for cell building block, so cell can grow or proliferate rapidly. Cancer cell increase the process of Barber effect or altered cellular metabolism, by which they produce more and more intermediates for cell building blocks. Another feature is cancer cell turn almost immortal because they have limitless replicative potential. Another feature is sustained angiogenesis. As we know, a growing tumor mass requires oxygen and various intermediates to grow faster and excretion of the harmful substances. They need more and more blood supply. Next feature is metastasis and invasiveness. This is one of the most peculiar feature of malignancy: metastasis to distant tissue, distant site, or invasiveness to the surrounding tissue. The last feature of cancer cell is ability to evade host immune system. In order to survive, they develop some special features, or by various mechanisms they evade the host immune system. So these 8 are the features of cancer cell. Cellular and molecular hallmarks of cancer cell. First is self-sufficiency in growth signals, in which we discuss proto-oncogene mutation. Second is insensitivity to growth inhibitory signals. In this we discuss about the tumor suppressor gene in their mutation. Third is evasion of apoptosis, and the cancer cell develops some altered cellular metabolism like Warburg effect. Cancer cell render immortal by limitless replicative potential. They have sustained angiogenesis for more and more oxygen supply and nutrient supply and removal of the harmful substances. Metastasis and invasiveness, and last is ability to evade host immune system. Hello everyone. Now we are going to discuss about another feature of cellular and molecular hallmark of cancer is evasion of cell death. How cancer cell evade cell death or escape cell death. So evasion of cell death. In normal cells, pro-apoptotic gene like BAC, they are pro-apoptotic genes or protein. They stimulate creation of mitochondrial pore and releasing of cytochrome C into the cytoplasm. So this is the mitochondria, and these pro-apoptotic proteins BAC will result into the creation of pore in mitochondrial membrane and releasing of the cytochrome C into the cytoplasm. Once this cytochrome C release in cytoplasm, this cytochrome C react with AFAB1 apoptotic activating factor 1 and result into the activation of caspase 9, which is responsible for the activation of intrinsic apoptotic pathway, ultimately result into the activation of executioner caspases like caspase 3, 6 and 7 and result into the apoptosis or cell death. So in the normal cell we have seen how pro-apoptotic gene stimulate the apoptosis or will result into the apoptosis. There are some anti-apoptotic proteins. They basically resist apoptosis or result into the increased cell life or cell survival. So anti-apoptotic proteins like BCL2, BCLXL, they are anti-apoptotic. And there is a third category of BH3 only proteins. They are the regulator of apoptosis, namely BIM, BID, BAD, PUMA and NOXA. They neutralize the anti-apoptotic proteins and will result into the apoptosis. So this is the scenario for normal cell. Pro-apoptotic gene stimulate apoptosis. BH3 only proteins like PUMA, they neutralize anti-apoptotic proteins and result into the apoptosis of cells with injury or irreversible injury. Now, in cancer cell or malignant cells, they have two mechanisms to avoid apoptosis. Cancer cells has two mechanisms to avoid apoptosis. First mechanism is loss of function of p53 gene. As we know, p53 is a tumor suppressor gene. Due to the loss of p53 gene function, this prevents upregulation of PUMA gene, which is a BH3 only gene, and which ultimately neutralize the anti-apoptotic protein. So due to the loss of function of p53 gene, this PUMA gene got neutralized, or sorry, this PUMA gene got no upregulation. So this ultimately prevent the apoptosis. No apoptosis, because PUMA gene has an inhibitory effect on anti-apoptotic proteins like BCL2 and BCLXL. And second mechanism is overexpression of anti-apoptotic members of BCL2 family. Overexpression of anti-apoptotic members of BCL2 family. This further decrease apoptosis and result into the cell survival. This is seen in follicular lymphoma and chronic lymphocytic leukemia. So in cancer cell, we have seen cancer cell develop two mechanisms to avoid apoptosis. First is by the loss of function of p53 gene. Due to the loss of function of p53 gene, there is no upregulation of puma gene, so there will be no apoptosis. And on the other hand, overexpression of anti-apoptotic members like BCL2 family proteins, and they ultimately result into the decreasing apoptosis, which is seen in follicular lymphoma and chronic lymphocytic leukemia. Another cellular and molecular hallmark of cancer cell is they have limitless replicative potential. So they have limitless replicative potential, or they have, we can say they have immortality. So now we will discuss in detail about this feature. Cancer cell have stem cell like properties through various mechanisms like evasion of senescence. Cancer cells do not get old. Second is evasion of mitotic crisis, and third is they have the property of self renewal like the stem cell. So the limitless replicative potential of cancer cell is due to the stem cell like properties of cancer cell, and this we will discuss under three headings. First is evasion of senescence, evasion of mitotic crisis in second, and third is self renewal. So the first is evasion of senescence. So what is senescence? In normal cells, after they serve their purpose, p53 gene and RB gene promote or stimulate senescent state transmission of the cell by various steps. But in cancer cell, as we know, p53 and RB gene mutated or mutation is there in cancer cell, so there will be no senescence and cells keep on living. The second feature, evasion of mitotic crisis. So what is the evasion of mitotic crisis? tumor cells stimulate telomerase activity or telomerase activity and DNA recombination. Both of this result into the increase cell proliferation. Tumor cell maintain the telomere. In normal cells, there is telomere shortening after every cell division, but in tumor cell there will be no telomere shortening. So in somatic cell, after division, after cell division, there will be telomere shortening or DNA damage. If p53 gene is present or normal, then cell will undergo senescent. If p53 gene is mutated, then this will result into the activation of non-homologous end joining repair activation. NHEJ pathway will result into the DNA repair or the formation of dicentric chromosomes. After dicentric chromosome formation, there are new double-stranded DNA breaks are there. Telomerase activity is there, then this will result into the cell proliferation or cancer formation. If there is no telomerase activity, then this will result into the mitotic crisis or mitotic catastrophe or result into cell death. So this is the evasion of mitotic crisis, in which we have discussed tumor cell maintain telomeres via increased telomerase activity or DNA recombination and result into the increased cell proliferation. And in somatic cell, we have seen after the cell division there is telomere shortening or DNA damage. If p53 is intact, this will result into the cell senescence. If p53 is mutated, then this will result into the activation of non-homologous end joining pathway activation. NHEJ pathway will result into the formation of dicentric chromosomes and new double-stranded DNA breaks. If telomerase activity is high, then this will result into the cell proliferation and cancer formation. If telomerase activity is low, then this will result into the mitotic catastrophe and cell death. Third mechanism of limitless replicative potential is self renewable. This is basically a property of stem cell. So cancer cell acquire properties of stem cellness through two mechanisms, or this cell renewal occur through two mechanisms. First is there will be stem cell cancer of stem cell itself. For example, chronic myeloid leukemia. And other mechanism is somatic cells acquire some mutation and transform into stem cell or acquired a property of stemness. This is seen in APML, acute promyelocytic leukemia. So we have discussed 2 points in this lecture, or 2 properties of cancer cell in their cellular and molecular hallmarks. First is evasion of apoptosis or evasion of cell death, and the second is limitless replicative potential. So this is it. Now we will meet in the next lecture. Thank you very much. Thank you for supporting. Sustained angiogenesis is to become a big mass or tumor. Cancer cell need oxygen and nutrient supply and removal of harmful substances. So these achieved by increase angiogenesis of that particular area or particular tumor. So how this achieved? Now we will see. There are some pro-angiogenic factors that we will discuss. First is hypoxia, lack of oxygen or relative lack of oxygen. Second is some driver mutation in tumor suppressor and oncogene. Driver mutations in tumor suppressor or proto-oncogenes. And third is proteases. So how this work, we will see one by one. First is a relative lack of oxygen or hypoxia. So how this hypoxia or relative lack of oxygen trigger angiogenesis? Hypoxia result into the stabilization of hypoxia induce factor 1 alpha. hypoxia induce factor 1 alpha. This HIF1 alpha induce or increase secretion of VEGF, vascular endothelial growth factor and fibroblast growth factor, and they will result into the formation of new blood vessels. The formation of new blood vessel is known as neovascularization. Tumor cell experience relative lack of oxygen and they stimulate the secretion of hypoxia induced factor 1 alpha and result into the neovascularization. Now we will see the mutation or driver mutation in tumor suppressor genes or proto-oncogenes. Now p53 is a tumor suppressor gene. Normally p53 promote thrombospondin 1 secretion, which is an anti-angiogenic factor. Or on the other hand, p53 inhibit VEGF. By inhibiting VEGF, also inhibiting neovascularization. And on the other hand, proto-oncogenes or oncogenes like RAS and MYC, they stimulate the secretion of VEGF and result into the increased process of or promotion of neovascularization. So this is the role of tumor suppressor and proto-oncogenes in neovascularization or angiogenesis. Third mechanism in sustained angiogenesis is the role of proteases. Proteases of two types basically: angiogenic and anti-angiogenic. Angiogenic proteases release B fibroblast growth factor, which is a pro-angiogenic factor we have already discussed. Release fibroblast growth factor from extracellular matrix. And on the other hand, anti-angiogenic proteases, they cleave plasminogen and collagen. Plasminogen is the cleaving of plasminogen will result into the formation of angiostatin and collagen cleaving result into the endostatin formation, and both of them are anti-angiogenic. So This is the mechanism of these are the mechanism of sustained angiogenesis in tumor cell. First, as we have discussed, tumor cell need more and more oxygen and nutrient supply and removal of harmful substance for increased growth. So there are three mechanisms: relative lack of oxygen or hypoxia induced angiogenesis, or mutation in tumor suppressor and proto-oncogenes like p53 and RAS and MEK will result into the neovascularization and role of proteases in angiogenesis. So this is angiogenesis. Another hallmark of cancer cell is evasion of immune surveillance. How they evade the immune surveillance? First, we need to understand the normal mechanism of immune system. A normal cell, after exposing to harmful substance or mutation, develop some antigen, new antigen, these antigens detected by immune cell as foreign antigen, and they eliminate the cell exposed to mutation. This is the normal functioning of immune system. Here we see some antigen or tumor antigen produced by oncogenic viruses. For example, Epstein-Barr virus produces Epstein-Barr virus antigen, HIBNA. Human papilloma virus produces E6 and E7 tumor antigen. So what are the anti-tumor mechanism normally our immune system have? First, we are going to see the normal immune system functioning against the tumor cell. Anti-tumor mechanism of our immune system. Sorry. Tumor cell identified by antigen presenting cell like macrophages and or dendritic cells, and they stimulate cytotoxic CD8 T cells and result into the elimination of tumor cell. This is the first mechanism. Second mechanism is by the involvement of CD4 T cells or helper T cell. Third anti-tumor mechanism is NK cells. They directly attack on tumor cells and result into the elimination of tumor cell. A fourth mechanism is T cells and NK cells secrete interferon gamma to activate macrophages and ultimately these activated macrophages eliminate the tumor cells. So these four are the anti-tumor mechanism of our immune system. First is antigen presenting cell identify tumor cell and activate the CD8 T cell. Second is by the action of CD4 T cell or helper T cell. Third is direct action of NK cell, and fourth via T cell or NK cell secrete interferon gamma and activate the macrophages for their anti-tumor action. So these are the four normal mechanism of our immune system. Now we will see the mechanism of immune invasion by tumor cells. Mechanism of immune evasion by a cancer or tumor cell. First is selective outgrowth of antigen negative variants. As we have discussed, after mutation, cancer cell develop some new antigen, but there are selective outgrowth of antigen negative variant. If there is no antigen, so it's hard to detect these cells and there will be no anti-tumor action. Second mechanism is loss or decreased expression of MSC class 1 molecule. So now the MSC class 1 is important molecule. If the expression of MSC class 1 is decreased, then they are hard to identified by antigen presenting cell, and that's why they escape the immune surveillance. Third mechanism is engagement of pathways that inhibit T cell activation. As we have discussed, CD8 T cell is anti-tumor in action, and CD4 T cell is also play an important role in anti-tumor mechanism. But in cancer cell there are development of certain mechanism by which they engage the pathways that activate the tumor cell and they inhibit them. Tumor cell increases CTLA4 and PD1 ligand. PD1 ligand bind with PD1 and they ultimately result into the T cell inactivation. How this occurs, we need to discuss about the normal activation of T cell. Normally, an antigen presenting cell have a MHC peptide which is identified by T cell receptor on the T cell. T cell receptor and a co-receptor known as V7 react with or attach with the CD28 of T cell, and this will result into the T cell stimulation. These stimulated T cell known as primed T cells, and they secrete some cytokines to harm the tumor cell. So this is the normal process. What in tumor cells, as we have discussed, they express PD1 ligand, program death 1 ligand, which act with the PD1 of tumors, PD1 of T cell. This PD1 got activated in T cell, and this PD1 inhibit T cell receptor. So there will be no engagement of T cell receptor with the MHC peptide of antigen presenting cell. Or on the other hand, as we have discussed, tumor cell also expresses CTLA4, which binds with B7 of antigen presenting cell and no T cell activation. So tumor cell inhibit T cell activation or suppress T cell activation by the action of two molecules, PD1 ligand, which inhibit the T cell receptor ultimately, and other is CTLA4, which bind with the B7 of antigen presenting cell and result into the T cell inactivation or inhibit the activation of T cell. So these are the anti-tumor mechanism like cancer cell depict. First is selective outgrowth of antigen negative variant. Second is loss or decreased expression of MHC class 1 molecule, and third is engagement of the pathways that inhibit T cell activation. Thank you.